All right. Well, thank you all for joining the Biosensors seminar series. We're super thrilled today to have Vatsan Raman. Vatsan is an associate professor at the University of Wisconsin Madison within the Department of Biochemistry. Vatsan has a really great background. So I believe he did his PhD with Dr. David Baker at the University of Washington, Seattle, where he contributed to the development of computational methods in the Rosetta biomolecular modeling software suite for predicting protein structures at atomic level accuracy. Following that, Vatsan was a Wyss Technology Development Fellow at the Wyss Institute, where he worked with Dr. George Church in his laboratory at HMS to develop foundational technologies to engineer microbes using biosensors to produce sustainably fuels and chemicals. So super excited to hear from Vatsan, who's going to be talking today about scalable, a highly multiplexed design of allosteric biosensors with new specificities.
All right. Well, thanks, Khalid. You guys can hear me okay?
Yes.
All right, perfect. All right, let's turn on my slide deck. Where did that go? I'm getting the dreaded rolling rainbow. Okay, perfect. This is fine. Um, this looks okay? Okay. Well, thanks, Khalid. Thanks for the kind introduction, and thanks for the opportunity to give a presentation here. So today I'll maybe tell a little bit about our work on designing allostatic biosensors. This is a problem that we've been interested in for many years. And as Khalid mentioned, the initial motivation for this was from the standpoint of microbial engineering, where we wanted to use these biosensors as cell-based tools to measure metabolite levels and to use these in conjunction with the host genome engineering to be able to sort out cells that are highly sort of productive for a certain target molecule. But then, as you can imagine, biosensors have applications which are much beyond bioproduction of chemicals. And so over time, we've sort of branched out in other directions. But I think one question we've always been fascinated with is this idea of, how do these allostatic proteins work and how can we co-opt them and redesign them to use as metabolite biosensors. And so this is sort of an abstract figure that basically tells you in general what my lab does, which is to figure out how sequence function landscapes work. So on the x-axis, you have some measure of sequence. These could be sort of protein sequences or could be sort of genome variants of an organism. On the y-axis, you have some measure of function. This could be activity, could be fitness, could be stability, whatnot. And so what we try to do is we try to develop methods where we can systematically map out the space for many biological systems and figure out what the fundamental rules are, right? And so one such class of proteins are biosensors, which are really sort of a cornerstone of synthetic biology, right? Because people have used these as I mentioned, in the context of metabolic engineering to use microbes as chemical factories, to accelerate DBTL cycles, where you can sort of use these to measure things and then sort of go through that cycle in a more efficient fashion. But these biosensors have been used to monitor metabolites within living cells as sort of knobs and switches in synthetic gene circuits. And I think there's a big push to use these as ex vivo biosensors in the context of using them as paper-based devices to measure, say, metabolites in the environment or sort of human fluids, or even as sort of portable wearable biosensors. So it's just a number of compelling applications. And in sort of, these allosteric transcription factors, which I'll get to in a second, and how they work, there's just a number of applications where different types of chemicals with different end uses, there is a need to sense them. So in the case of, sort of progesterone would be for sort of treatment guidance for, people who are, families who are trying to get pregnant. Fentanyl would be a case where you want to use these to detect the presence of illicit substances. In the context of biodefense, there are small molecules that are such as saxitoxin and other toxins that the DoD wants to sense. Food safety, aflatoxin, which is a fungal toxin found in contaminated food, bioremediation, and so again, there's a number of different application spaces where there's a need to sense metabolites. And we've been working with these fairly simple proteins. These are one component sort of signal transduction systems in bacteria. They're as simple as it gets. And the way it really works is that you have this ligand binding domain and a DNA binding domain. So this is a transcription factor that sits on DNA. And then when it binds to its small molecule of interest, it undergoes a conformational change, which is allosteric, and then it falls off DNA. What this then means is that you can then measure the activity of the presence of the molecule, of the inducer molecule, or the concentration of the inducer molecule by hooking up a reporter to the system, and then you can essentially read out the reporter and then infer the concentration or the presence of the inducer molecule. And so people have sort of used this for years and years. And what's really nice about the system is there is thousands and thousands and thousands of these out in the nature, right? So really, there's just an extremely large number of these that are out there. But it really turns out that even though there is a very large number, the biosensor and the ligand space is kind of sparsely mapped because just because there is sequence, something is identified as a known biosensor based on sequence similarity doesn't mean that we actually know what ligand it binds to. And so if we imagine this case where you have this on the x-axis, you have all possible sequences of allosteric transcription factor biosensors. Along the y-axis, you have some measure of the chemical space of known biosensors, right? This is sort of somewhat of a sparsely populated space. And so one motivating question for me in my lab has been, how can we sort of expand that space where we take each data point of a ligand transcription factor pair and then redesign these transcription factors to sense new molecules to sort of expand the repertoire of molecules that can be sensed and for which we can have biosensors. And so this has been a sort of an ongoing pursuit in the lab for a few years now. And people have sort of have done this with a few different approaches. One maybe classic way for designing biosensors for new molecules is to be able to hook up a reporter and then do fact sorting. So you design a protein, you challenge it with a small molecule, and then you have some GFP reporter. And depending on the activity of the GFP reporter, you can estimate whether your designed protein responds to your small molecule. And this is an approach that people have used for the last 10, 15 years. Now, there are some fundamental challenges with this FACS-based screening. So because although FACS screening is high throughput, there are some sort of key weaknesses that we found hard to overcome when we were using this. One is that the weak activity is difficult to detect. Often with design challenges, you don't get a runaway success the first time you design. You get something which has weak activity, and then once you're able to fish that out, you can then improve upon that through iterative design, right? But the problem with sort of FACS-based screening is that if you have something that shows twofold, threefold, fourfold activity, it's just hard to pick those out. So that's number one. Number two is that because allostery is this sort of baked-in property of these transcription factors, and oftentimes when you introduce mutations to these transcription factors, you break the allosteric linkage, which means that then the protein no longer binds to DNA or can become unstable. And these variants essentially take over the population because the successful designs are fairly sparse. You have these broken variants that sort of overwhelm the design, the experimental pool, and hence these dominate the screen. And the other problem is that when you run sort of this winner takes all kind of screens, you can evaluate, say, 1,000,000 variants, and let's say you have 10 winners, right? The problem is the rest of the information is basically lost, right? And that's useful information of things that don't win the screen, because then that tells you if you were to do a second generation design or a third generation design, you want to know which sequences worked and also nearly as importantly, which sequences did not work. And finally, if you want to run, say, a pool of ligands, sort of a library of designs on a number of different ligands, this traditional sort of FACS-based approach simply does not scale. And so what we started sort of thinking about, how do we, improve the design methodology by which we can sort of create these sequences that are likely to be novel biosensors, and B, how can we then modify the screening methodology so that we can pick out sequences with high sensitivity and weak activity among a large population of potential, library variants. So if you sort of think of this as sort of sort of a direct evolution problem. So the question is that you start with a starting sequence and you sort of walk through the sequence of fitness landscape to get to some optimal sequence minimum. And one way is how do you introduce, what sequences do you want to have as you diversify your starting scaffold? And so this is work that we did in collaboration with Fleischmann Lab at the Weizmann Institute, where Sorel Fleischmann had this really simple method where the idea was that let's say you have a population of so in this case this is a transcription factor on the right hand side. You can create a multiple sequence alignment of the transcription factor, hundreds or maybe a few hundred sequences. And based on this, the multiple sequence alignment, and let's say you focus on the amino acids that are part of the binding pocket, right? So what you can create is this phylogeny-guided sort of position-specific scoring matrix, right? Along the x-axis here, you have the amino acid identities. Along the y-axis, you have position numbers. The blue positions are not conserved, right? So based on the multiple sequence alignment, the red positions are highly conserved. So what you can then do is you can say anything that is not conserved below a certain level, right? So it's highly variable. It doesn't really matter what the amino acid is. I'm just going to completely ignore that set, right? So based on that kind of a, sort of a cutoff, you can remove everything that is not conserved, and you're left with a smaller set of designable sequences. And once you have the smaller set of designable sequences, even with this smaller set, the combinatorial space is fairly large. And so now what we then do is we use Rosetta to then ask which of these mutations, if introduced into my scaffold of interest, does it destabilize that mutation? So in other words, you take the information from the phylogeny of sequences of that family, but then you put specific mutations into that scaffold and ask whether it's compromises the stability of that protein or not. And once we go through that process, and so this list gets even shorter, and so now we're left with an even smaller list of positions that are relatively conserved, not super conserved, but relatively conserved, and mutations that don't destabilize the protein, right? And so once we have this minimal set of mutations, then we can, and so these are the positions. So for instance, in the case of TTGR, which is a TET-R family transcription factor. So these are obligate dimers. And so here are the positions around the ligand binding pocket, the residue positions, and these are the allowable residues based on the criteria that I described in the previous slides. And so once you take these allowable residues, you can start making combinatorial libraries of these different mutations. And then we generally restrict it to no more than 4 or 5 mutations per sequence. And so that combinatorial space of sequences based on these amino acid changes is about 17,500 sequences. Okay, so now we have some initial set of sequences that we've identified based on the rationale that I just described. Now the question is, well, how do we evaluate these sequences in a pooled fashion? I already told you what the drawbacks of using FACS is, and how can we sort of whatever this new method is, we ought to be able to pick out these weak hits, right? So that's clearly one important criteria. And so to do this, we wanted to be able to read out the activity of the transcription factor using a sequencing-based readout. And the advantage of using a sequencing-based readout is that it's highly scalable, it's highly sensitive, and simply by scaling up the volume of sequencing, you can really amass a lot of data that can tell apart even small functional differences, right? And so the question is, so how do we take a transcription factor and then turn its activity into a sequencing-based readout? And so the way we did that was to say, here is on the transcription factor on the left-hand side. Instead of producing a fluorescent protein, all that we did was to say, let's produce a transcript with an RNA barcode, right? And so we're not looking at a translational output, we're looking at a transcriptional output. And the advantage of using a transcriptional output is that you can barcode the sequence, right? You can barcode the RNA. And once you can barcode the RNA, you have to establish a relationship between the RNA barcode and the transcription factor variant, right? So you have to know which RNA barcode corresponds to which transcription factor variant. And all that you then need to do is simply sequence the RNA barcode before and after a ligand challenge, and that'll give you, in a single pot experiment, the function of thousands of transcription factors in one go. And I'm happy to sort of unpack the details here for if you have any questions, but I don't want to spend too much time on the method. But the idea really is to be able to link the activity of a transcription factor to a barcoded RNA, and then measure the ratio of the RNA levels plus ligand versus minus ligand. And that ratio is the activity of the transcription factor. And so the way we would do this is you would have sort of an oligo pool library of transcription factor variants, and then we would have a reporter which has a barcode that is attached at the very end. And then we would create this mapping between the variant, transcription factor variant, and the barcode. And there's just some little cloning trickery that has to happen to be able to establish that relationship. And once you establish that relationship, here is how that might work. Let's say you take this blue transcription factor, you diversify it, and let's say there are 3 different variants, right? one variant that does not produce too many transcripts. So that's this guy down here. Then you have this other guy up here, which produces some intermediate level of transcript when it challenged with the ligand. And here's the yellow guy that produces a ton of transcripts when you challenge it with the ligand, right? So what this means is that the yellow guy is the guy that has the strongest response to your ligand challenge, and you can do cDNA synthesis and you can sequence it and you can quantify that effect. So that's how that works. And so we first wanted to ask, is this idea, does it even make sense? Can we do some clonal validation to check if this pooled RNA-based sequencing method, which we call sensor-seq, does that work? And so we did some, qRT-PCR measurements on clones, and then we did some summed barcode measurements, just again to compare the pooled measurements versus clonal measurements, and we saw some reasonable correlation, which just made us think that we can now move forward with the idea of running a much larger library. And so this is, again, about 17,500 design sequences for TTGR. This is within the ligand binding pocket, and I'm just sort of pointing out the key amino acids that are contacting the native ligand, in this case, naringenin. And so for the initial design, we chose naringenin was one of the targets. It's just because it's a native ligand and it sort of provides a good positive control. We chose a couple of tamoxifen prodrugs. And so these are drugs that are used for breast cancer treatment. We chose quinine, which is an antimalarial drug. We chose naltrexone, which is used as an opioid antagonist when people overdose with certain types of opioids, and ellagic acid, which is an antioxidant. And these molecules are basically chosen based on some Tanimoto score distance. These molecules were not previously exposed to TTGR, so we said, we don't want it to be too close to the native ligand, but at the same time, we didn't want to completely sort of jump off the cliff and pick like some short linear molecule that has no chance of sticking to TTGR. So we just sort of balance out the distance from the starting ligand to sort of move farther away. And so we initially picked the set. And so this is what the data looks like. Each line is a unique sequence, right? So there are 17,500 sequences in this heat plot. The more maroon it is more activity. The more sort of blue it is less activity, and zero is sort of like wild type. And so what you'll see here is that about half the sequences that we have in the set respond to naringenin, which is not totally surprising. So we did not design away from the native ligand. We just designed toward other ligands. So about half the sequences show response to the native ligand. Now, this is the result with tamoxifen. Again, you'll see that there's a lot of sort of white there, which means these don't respond to tamoxifen. But you'd see about 20 to 25% of sequences do respond to tamoxifen. And here is 4-hydroxytamoxifen, which is only different by a single hydroxyl group between tamoxifen and 4-hydroxytamoxifen. And again, this has a slightly different profile from tamoxifen. But what's kind of neat is that now we can start comparing these sequences, and you'd say, for instance, the top group responds to both tamoxifen and 4-hydroxytamoxifen. So these are bispecific sequences. The second block here only responds to tamoxifen, but does not respond to 4-hydroxytamoxifen. And the last block down here responds to 4-hydroxytamoxifen, but not tamoxifen, right? So you kind of get these specificity profiles for free when you run these pool selection experiments. Now you can look at quinine, which is yet another molecule. And then in this case, we were actually quite surprised that we found more than 50% of sequences did respond to quinine. Now, in the case of naltrexone, this was really quite remarkable because you'll see that almost the entire sequence library population do not respond to naltrexone, right? But then there's little sliver of sequences that do appear to respond to naltrexone, and I'll show you sort of data that actually validates this. But what's nice is that this is less than 5% of the population, right? But the fact that we've been able to pick out these sequences from among 95% of sequences that don't respond was really promising for us, that the method is sensitive enough to be able to fish out these weak activity guys that one could then go back and re-optimize for better activity. And so that we had achieved a level of sensitivity that would allow us to pick out weakly active de novo hits. Now, ellagic acid is another case where we had appeared to have weak activity, but this is a case that was actually eventually turned out to be a failure because although we did see what appeared to be weakly active hits, they did not, sort of when we did the clonal validations, they did not work out. So this would be a case where the design did not work. And also once we do this initial mapping, we'd say, okay, there is a certain false positive rate with these large scale screens. And so what we did was we'd say, give me the top 50 design sequences based on this initial sensor-seq-based pooled scoring. And then we would resynthesize those top 50 sequences and now put them through a GFP, sort of more of a traditional GFP-based screen. So in this case, this is quinine. This is the top 50 design for quinine. This is in the absence of quinine on the left-hand side, and the right-hand side is presence of quinine. And you see the sort of dramatic right shift with GFP. And so that tells us that, we've indeed sort of enriched for successes for the quinine based on more of a traditional GFP-based activation assay. And so now we can then pick out the top 15, 20 sequences for each ligand, and then we can clonally validate them against all the ligands. And so this is what that specificity profile based on clonal validation looks like. The 6 ligands are up here. These are the 2 native ligands. And so what you'll see, for instance, is that there are certain sequences that only respond to naltrexone, but not to the others, right? So you see dark blue for naltrexone, but not the others. Other sequences that only respond to quinine. But then once you get to this guy 2G9, it responds to quinine and N-desmethyl tamoxifen, right? So there's some broader diversity at some sequences. Now, here are sequences that appear to respond to all 5 ligands, but not naltrexone. And here are sequences that respond to N-desmethyl tamoxifen and tamoxifen, but not the other types of tamoxifen variants. And so what's nice is that you sort of learn these rules of specificity, not because we're designing towards specificity, it's just that different sequences have different specificity profile. And the fact that we've been able to measure them in a pooled fashion, we can read out these different specificity profiles for free from our data set. Now, it also turns out that most of these ligands, most of the sequences do retain the ability to respond to the wild-type ligand naringenin and fluoretin. And I think part of the reason for that is we did not do negative design. It's hard to lose wild-type activity. One has to explicitly design against the wild-type activity in order to kill that, and we just did not do that. And I mean, that's an obvious thing that we want to try going forward, is to ask, can we design towards new function, but also sort of jettison the wild-type function towards the initial native target molecule. And another sort of fallout of these large-scale data sets is that we can now start looking at enrichments of specific amino acids at specific positions and what that might tell us about the molecular basis of specificity. So for instance, in this case, we have different amino acids that were part of the library along the x-axis at different positions and the different ligands on the y-axis. And so this essentially tells you what is the enrichment of a certain amino acid post-selection on a particular ligand compared to the initial library, right? So did a certain amino acid get enriched or did it get depleted after, say, selection, right? Sorry. So in this case, so let's say, for instance, we can focus on, say, 4-hydroxytamoxifen and tamoxifen, and you'll see that, the profile is largely similar, but then you can start seeing there are certain amino acids that were depleted, there are certain amino acids that were enriched between these 2 groups, which then tells you that these key changes are likely the determinants of specificity between 2 fairly similar ligands, right? And you can then say, again, look at naltrexone, which is a completely different ligand. Again, the enrichment profiles of amino acids for naltrexone are very different from the other 2 ligands. And so again, you end up learning what are the key drivers of specificity between different small molecule targets. And so we really wanted to sort of make sure that we were indeed designing proteins that actually bound to the molecules of interest. And so we ended up getting a crystal structure of TTGR bound to naltrexone. And so these are the 4 amino acid substitutions in the design sequence: L70A, 89W, 106M, and 110F. And this is naltrexone. And just it's worth noting that naltrexone is a completely synthetic molecule, right? And this TTGR is this ancient transcription factor which has never seen naltrexone before. And so here's a crystal structure of naltrexone bound to this TTGR. And we can sort of look at the crystal structure and then rationalize why this might be happening. The opening has become a little bigger, and there are key amino acid substitutions that we think enable TTGR to bind to the small molecule. And so now we're at the point where we've done this initial screen, but because it's screening a larger molecule set is fairly trivial, it's just a little more sequencing for us. Now we've actually gone off and screened about 15, 20, 30 different molecules. And the success rate has been about 60-65%, where we've been able to find hits for a whole bunch of new molecules. So this is not the exhaustive set, but these are some of the molecules that a couple of my students are looking at. And I'd say about for 60% of cases, we have either at least a weakly active biosensor or something that's doing better than that for some of these molecules. And so now that we have this large data set of sequences against ligands, so 17,000 sequences against 50 different ligands, that's a fairly large data set. And so on the one hand, you have protein sequences, which are the variants that we have in our population. And then on the other hand, we have the ligand structures, which are sort of chemical representations of different ligand sequences. And we can start building a model to ask, what is the relationship between different mutations and different sort of more granular descriptors of ligand structures. And the goal there would be to start having a predictive model so that if you now give me an arbitrary ligand sequence, I can, I should be in a position to at least come up with some potential protein sequence candidates that might be able to bind to this target ligand. And so that's what we're working on currently, is to be able to build this model to be able to design biosensors for an arbitrary target. So the key takeaways is that the sensor-seq methods can scale quite trivially to many ligands and many protein scaffolds. We haven't done many protein scaffolds, but that's the direction that we're headed in. We have a rich data set for machine learning where we can then design biosensors for target ligands of interest. What's kind of remarkable to me as sort of a biochemist is that these allosteric transcription factors are evolvable towards completely non-native molecules. These are molecules that they've never seen before. And it tells you something about the evolutionary malleability of these proteins. And I guess one sort of interesting question is, why are some proteins are more evolvable towards new molecules than other proteins are? So what is a fundamental property of that of a certain protein that makes it more amenable to being evolvable than, say, other candidates? And so with that, this work was funded by the US Army. And here are people in the lab who do all the cool stuff.